Hi guys. So first I want to apologize for getting this video to you late. I've been having some issues with my laptop as well as with Microsoft that was making it difficult to get this recorded the way that I needed it to. Because I am getting this to you, so I am going to give you an extension for the Lab 2 assignment. It'll be due on Saturday at midnight instead, so that you guys have a few extra days in case you have any questions. I have finished reading through all of your entry surveys, and I've started grading your Lab 1 assignments, and I am really excited for the semester. You guys seem very open-minded and very excited to learn, and you guys have a very wide range of interests, which I don't think we'll be able to get through in just this one semester, but hopefully we can at least touch on most of them and answer some of your questions. But we're going to start today with talking about genetics and inheritance, specifically about patterns and mechanisms. So some key terms that you need to know. A Punnett square, you've probably done some of these if you ever took a biology class in high school, but they're just a method of diagramming inheritance where parent genotypes are used to estimate the probability of various genotypes and potential offspring. So you're using parent genotypes to determine potential offspring genotypes. You also need to know what an allele is. It's just an alternate version of a gene that is going to code for these different traits. These alleles can be dominant, which you can see with this uppercase B. These dominant alleles mask the effects of recessive alleles. Recessive alleles, like this lowercase b, are masked by dominant alleles. So they're going to be covered up by these dominant alleles. A gene is just a section of DNA that codes for a specific trait. A genotype is the specific alleles that an organism has for a trait. So that's going to be your double uppercase B's or your double lowercase b's, or your combination of the two. A phenotype is the physical expression of a genotype. So your genotype is going to be the actual letters that you're using to represent those alleles that are coding for a trait. Your phenotype is how it actually looks. So it might code as a genotype for that double capital letter B, and maybe that B means that blue is the dominant gene. Lowercase b is white. That double BB is always going to show up as blue because there's no recessive alleles. When you have a heterozygous allele or genotype, it's going to be a mix of the two. So one dominant and one recessive. You can see that as a capital B and lowercase b. The genotype for anything heterozygous like this is going to be the same as the phenotype for anything heterozygous like this is going to be the same as the phenotype for anything that is homozygous dominant. When you have a homozygous dominant, homo means one or the same. So you know that it's going to be two dominant alleles for the trait, so two uppercase letters. If it's homozygous recessive, you're going to have two recessive alleles for the trait or two lowercase letters. And I'll show you more about how to do these here in a second. But we get all of this from Mendel. Mendelian genetics, Mendel's work with these pea plants, he created this blending model, and he named the mechanism for this particulate inheritance. To do this, he had to use true breeding pairs. You can see here in the little picture that P generation, that's your true breeding pairs, which means that one parent is going to be homozygous dominant and one parent is going to be homozygous recessive. This is done because it reduces the chance of interference from things like codominance or incomplete dominance, which we will talk about in the later slides. But here in this picture, which is the same one that's on your guys' homework, you can see that P generation or parent generation, they're all tall and all short. So it's going to be capital TT, lowercase tt. Then you get to this F1 generation, or the first filial generation, and this is the offspring of the parent generation. So when you move over here, this one is not correct, but just for reference. These are going to be, let's see if I can, these are going to be that P generation, and everything in this box is going to be your F1. Then you just do the same exact thing for the F2. When you do the Punnett square using the F1 generation as the parents, then everything that's within those boxes are going to be the offspring, or the F2 generation. You can see this a little bit better here. We've got this P generation with one homozygous dominant and one homozygous recessive. They're using T's and S's for this. You can do them both as T, just with an upper and lower case. You can do totally different letters. Most people try and keep it kind of related to the trait using like the first letter of whatever the trait is, but you don't have to. But you can see we're going to take this homozygous dominant parent and put it up here, and this homozygous recessive parent and put it down here. You can flip-flop these, it doesn't matter which side they're on, but they need to stay together. So your genotype for your parents need to stay on the same side of your Punnett square. Now, whenever you are completing this Punnett square, you're going to take your dominant one up here, or wherever. If it's on the side, that's fine too. And you're going to combine it with the one on the side. Now, because these are homozygous dominant and homozygous recessive, all of the offspring are going to be heterozygous. These offspring are then your F1 generation. They are going to be all tall for their phenotype, because even though their genotype is heterozygous, that uppercase T still masking that lowercase s. So they're all tall, heterozygous. Whenever you move on to do this second generation, you're going to move some of the children from here, and they're going to become your parents for this Punnett square. Everything in this Punnett square is now going to be your F2 generation, because it is their offspring. And you're going to do the same thing. So you're going to read this. You can go down and then to the side. You can go to the side and then down. What you need to remember is your dominant allele in these instances needs to come first. So with this one, we're going to have TT with a dominant, or homozygous dominant, sorry. Then you're going to have two of these heterozygous. If you follow it down, you've got that T, and then that lowercase s from the side. Whenever you combine these ones going this way, you're going to have homozygous recessive. You can also see in this one, those are your genotypic ratios. So for this F2 generation, you're going to have one homozygous dominant, or TT, one homozygous recessive, or SS, and then two heterozygous. So you're going to show this for your genotypic ratio as 1 to 1. You're going to do it this way because we tend to want to keep that recessive allele the furthest down. So you want to start with your most dominant and end with your most recessive. This right here, that is your P ratio, and it's going to be 3 to 1. This might be kind of confusing because obviously there are not 3 of the same genotype. But whenever you're doing phenotypic ratios, you don't really care what the genotypes are. You care what they are coding for, what we can actually see. So even though this heterozygous and homodominant are different genotypes, they're still going to code for this plant being tall. Whereas, let's see if I can change the color of this. This homozygous recessive is going to be short. So in terms of what we can actually see, 3 of them are going to be tall, 1 of them is going to be short. It's easier when you have things like this P generation over here, because everything is the same. So your G ratio is going to be 1. Oh, that's not what I wanted. It's going to be 1 to 0, because there is only one genotype present. You can also think of these as ratios, so you could do them out of 100. Obviously, for this P generation, it's going to be 100 out of 100. Or you can do them in fractions. For this one, all 4 are that heterozygous. For this one, you're going to have 1 out of 4 be TT, 1 out of 4 be that homozygous recessive, and then 2 out of 4 be heterozygous. What's really important, I don't care if you guys do it by percentage or if you do it by fraction, whatever makes sense to you is fine. You do need to write them as a ratio. So they need to be in this form for your final answer. Oh, and I guess for this P generation, I should also mention that because they're all the same genotype and the resulting plants are going to be all tall, your P ratio is also going to be 1 to 0. You can also have other types of inheritance, like we mentioned. Incomplete dominance is when a dominant allele can't completely mask the effects of a recessive allele, and the resulting offspring have a physical appearance of a blend of both alleles. So in this example, you can see a red flower and a white flower. Red is dominant to white, but it can't fully mask the white. And so all of the offspring that are heterozygous are going to be pink because the phenotypes blend so that you see a combination of the two. You can also have codominance. Be careful not to mix these two up. With codominance, neither allele is really dominant or recessive. They can't mask each other, and so both are going to be expressed in the offspring. But they're not going to blend like they do with incomplete dominance. So in this example, you can see a white bird and a blue bird. Their offspring is going to be blue with a little white belly because neither color can mask the other one, but they're also not blending. So that's the difference. With incomplete dominance, the dominant gene is still trying, or allele is still trying to mask the recessive. It just can't fully do that. And so it blends the colors or the traits. Whereas with codominance, neither one is dominant or recessive over the other. And so they're both expressed. If we go back to the flower example, instead of the offspring being pink, they might be white with red splotches or stripes on them, or red with white splotches and stripes on them. So just be careful not to get confused on those. And then I did want to talk a little bit more about Punnett squares. So your guys' Punnett squares, most of them are single trait, so they're pretty easy, and we've kind of covered them in this lecture. But you're also going to have a dihybrid Punnett square, and that's where you are doing a Punnett square for two traits. Punnett squares can obviously get much more complicated with multiple traits, but it's unnecessary for you guys to have to do more than just a dihybrid. All right. So for this one, we're going to have one parent that is homozygous dominant for a trait. These L's are going to be long, lowercase is going to be short, the uppercase Y is going to be yellow, and lowercase y is going to be green. We're just going to keep the same concept of the pea plants. We're going to have one parent that is homozygous dominant, then heterozygous, and then one that is homozygous recessive, and homozygous dominant. Okay. Please ignore how ugly my Punnett square is going to be. Not used to drawing on this little pad thingy. Oh, they're pretty uneven, but that's okay. So you're going to follow the same rules that you do with a normal Punnett square. It's just going to get a little bit more complicated whenever you are designing your actual Punnett square, because you can't just lump these in any random way that you want, and you can't just put one letter on each row or column. Instead, you're going to start with your first letter, and you're going to go through and combine it with both of these second letters. So we're going to have capital L, capital Y, capital L, lowercase y, and then you're going to move on to the second L and do the exact same thing. So in this case, we're just going to repeat, and you're going to do the same for this. Because they are all the same, you're just going to have L, Y, L, Y, L, Y, and L, Y. Whenever you're filling your boxes out, it's going to be the exact same as a normal Punnett square. You want to start with this dominant gene. These are both capital Y's. Because all of this is the same, you know that all of this is also going to be the same. So just follow this down. They're all going to be heterozygous for height and homozygous dominant for color. Doing the same thing over here. Now notice, just because this L is not dominant to these Y's doesn't mean that I split it up. You want to keep things that are coding for the same traits, that are the genotypes for the same traits, together. These are going to be the same. This is also going to be repeating. And they're going to be the same going down. That's not what I wanted. Okay. So from this, we're going to try and do our genotypic ratio. And our phenotypic ratio. Right. So we know this is the same, and all of these are the same. If we move on, these are all the same, and these are all the same. So for our G ratio, there's only 2 resulting genotypes in this Punnett square, which means that you have a 50-50 chance. So it's going to be 1-to-1 for your ratio. For your P ratio, we can look at, and you don't have to write these out if you've got it figured out, but it could be helpful. We're going to look at our resulting phenotype or genotypes and figure out what they're actually coding for. So for this one, we've got heterozygous for length, so it's going to be tall or long, whatever you want to call that. We'll do long because I said that originally. And we're going to see that homozygous dominant for the color. So it's going to be long and yellow. Now for this resulting, where they're both heterozygous, it's going to be long and green. Oh, nope, sorry. Also yellow. I'm not paying enough attention. Be careful of that when you're doing these on your own. So our P ratio is actually just going to be 1-to-0. Now, if we change the second parent to something else, let's say parent 2 is actually going to be homozygous recessive. So we're going to have all lowercase l's. And homozygous recessive for color. Oh, no, we already did that. Oh, yeah. Here we go. So we're going to have L, 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 all recessive. All right. So now, this is going to be long. And instead of yellow, the resulting phenotype is going to be long and green. Well, now your phenotypic ratio is going to switch to being the same as your G ratio as a 1-to-1. They can get more complicated depending on what your parents' alleles are. You might have p ratios that end up being like 6,622 or all kinds of different combinations. But just be careful whenever you're completing these dihybrid Punnett squares that you're looking at all of the squares that you're doing, because it's really easy to make a mistake or overlook something. That is all I have for you today. Let me know if you guys have any questions, any concerns, and have a good rest of the week. Try and stay out of this upcoming cold weather.